DNA replication is a critical biological process in which a cell copies its DNA before cell division, ensuring that each daughter cell receives a complete set of genetic information. This process is semi-conservative, meaning each new DNA molecule consists of one original strand and one newly synthesized strand. Here's a step-by-step breakdown. 1. Initiation. 1. Origin of replication. DNA replication begins at specific locations on the DNA molecule called origins of replication. 2. Helicase unwinds DNA. The enzyme helicase unwinds the double-stranded DNA, creating a Y-shaped structure called the replication fork. 3. Single-strand binding proteins. These proteins bind to the separated DNA strands to prevent them from reannealing, reforming a double helix. 2. Priming. 1. Primase. This enzyme synthesizes a short RNA primer on each template strand. 1. The primer provides a starting point for DNA polymerase, the enzyme responsible for synthesizing new DNA strands. 3. Elongation. 1. DNA polymerase. This enzyme adds nucleotides to the 3' end of the RNA primer, extending the new strand of DNA in a 5' to 3' direction. 1. DNA polymerase reads the template strand in the 3' to 5' direction. Leading strand. The strand synthesized continuously in the same direction as the replication fork. Lagging strand. This strand is synthesized discontinuously in small segments called Okazaki fragments since it moves in the opposite direction of the replication fork. 4. Okazaki fragment joining. 1. DNA ligase. Once the RNA primers are removed and replaced with DNA nucleotides, DNA ligase joins the Okazaki fragments together to create a continuous strand. 5. Termination. 1. Replication completes. Once replication forks meet or reach the end of the chromosome, the process ends. 1. In eukaryotes, telomeres, repetitive sequences at the ends of chromosomes, play a role in managing the completion of replication for linear chromosomes. Key enzymes involved. 1. Helicase unwinds the DNA double helix. 2. Primase synthesizes RNA primers. 3. DNA polymerase adds nucleotides to synthesize new DNA. 4. DNA ligase seals gaps between Okazaki fragments. 5. Topoisomerase relieves the tension caused by unwinding of DNA. Accuracy and proofreading. DNA polymerase has a proofreading function, correcting most errors that occur during replication, ensuring high fidelity in copying the genetic material. This entire process is essential for cellular growth, development, and reproduction. Errors in DNA replication can lead to mutations, which may have various biological consequences.